my name is Mandela McGall, and I'm going to be presenting an experiment called the effect of temperature on the activity level of the enzyme trypsin. This experiment was performed by myself and my group mates, Adelina Minter, Carla Hernandez, David Padrowski, and it was done in coordination with Towson's Department of Biology. So, to introduce this, enzymes are proteins that catalyze chemical reactions in living organisms. Without enzymes, the chemical reactions that sustain life would not be able to occur at a fast enough rate. Molecules that need to be catalyzed are called substrate molecules. Substrate molecules bind to enzymes, and enzymes lower the activation energy by breaking bonds in the substrate molecule. When the substrate molecule initially binds to the enzyme in question changes shape, which then causes the change in shape of the substrate molecule, which then lowers the activation energy of the reaction. With the activation energy of the reaction lower than it would be without the enzyme, reactions are completed at a faster rate. Trypsin, the enzyme that we're talking about here, is found in the small intestine. It digests dietary protein by catalyzing the hydrolysis of peptide bonds, resulting in individual amino acids or small peptide chains. Trypsin is active in the lumen of the small intestine, where the pH is about 8. Trypsin, like any enzyme, has a range of optimum temperature where it is the most active. Changing the temperature so that it's too high or too low results in the trypsin not being able to break down proteins. So we asked, how does temperature affect the rate of the enzyme activity in the reaction between trypsin and the substrate azocasein? Specifically, we hypothesized that as the temperature increased from 37 degrees Celsius, enzyme activity will increase until it hits its optimum temperature of 50 degrees Celsius, where it will decrease from there. Trypsin exists in the human body, which has a normal temperature of 37 degrees Celsius. So this is why we believe that the enzyme activity will increase from 37 degrees to 50 degrees Celsius. And then after which the temperature will be too high, the enzyme will begin to denature, which would then reduce its overall activity. So moving on to materials and methods. First, 450 microliters of reaction buffer with a pH of 8, sodium chloride concentration of 0.3 molar, and 20 mm tris HCl were placed into each treatment. Then 500 microliters of 0.5% azocasein in deionized water was added to each treatment. Following that, 50 microliters of trypsin, that was 2 milligrams per microliter in a solution, with a sodium chloride concentration of 0.15 molar, pH of 8, and 20 mm tris HCl were added to the treatments. The treatments were then incubated in temperature-controlled water baths at 37, 50, 60, 70, and 80 degrees Celsius for 20 minutes each. After incubation, 500 microliters of 10% TCA were added in each treatment to stop the reaction of trypsin with azocasein. All treatment tubes were then centrifuged at maximum speed for 2 minutes. After centrifugation, supernatant from each tube was transferred into a clean cuvette. The absorbance of each supernatant was found at 440 nanometers after blanking the spectrophotometer with the buffer solution. So, continuing our materials and methods, as stated previously, TCA was added to prevent the trypsin from reacting any further and breaking down any more azocasein. The tubes were centrifuged to separate the pellet, which was the undigested azocasein protein, from the supernatant, which was the buffer mixed with the sulfanilamide groups released from the breakdown of polypeptides bonds in the azocasein. Sulfanilamide is an orangey color, so the more that is released, the darker the supernatant would become, thus having a higher absorption at 440 nanometers. The control variables of this experiment were a pH of 8 and a salinity of 0.3 m sodium chloride. These were chosen due to their similarity to the internal conditions of the human body, thus having little effect on the enzyme activity outside of its optimum range. 3 negative controls were used at the temperatures 37 degrees Celsius, 50 degrees Celsius, and 80 degrees Celsius. In the negative controls, 50 microliters of water were added in place of the trypsin. These were added to distinguish the very minimal amount that trypsin breaks down on its own, so it can be compared to our values in the end. So, moving on to the results, all shown here. Shown here is a table of the absorbance values recorded at each temperature. 3 replicates were taken at each temperature so the variability could be measured and any outliers could be identified. The replicates were then averaged and the averages were recorded: 0.349 at 37 degrees, 0.618 at 50 degrees, 0.555 at 60 degrees, 0.351 at 70 degrees, and 0.285 at 80 degrees. The standard deviation was also calculated for each temperature, shown here, and in each case it was low enough to assume that the average accurately reflects the data. From there, a bar graph was created of the averages with the temperature on the x-axis and the absorbance on the y-axis. It is clearly shown on this graph that the max absorbance was recorded at 50 degrees. It's our longest bar. After this, a t-test was also performed for every single data set, which compared the average absorbance at 50 degrees to the average absorbance of each other temperature. The null hypothesis for each, stated here, is that there would not be a significant difference between the absorbance values. The p-values are also recorded in this table, each of which fell under 0.05, suggesting a significant difference in each case. As such, the null hypothesis was rejected for each case. So, moving onward to our discussion. Our experimental design was suitable for our experiment and enabled us to accurately test our hypothesis that enzyme activity will reach its optimum point at 50 degrees Celsius. Our results support our hypothesis. From our results, we were able to determine that the most enzyme activity occurred at 50 degrees Celsius, because we recorded an average absorbance value of 0.618, which was the highest recorded. Our t-test also showed that there is a significant difference between enzyme activity at 50 degrees Celsius and all the other temperatures. Group A from Lab Section 9 also tested the effective temperature on enzyme activity. They tested the temperatures at 37 degrees, 43 degrees, 50 degrees, 57 degrees, and 63 degrees Celsius, and also found that 50 degrees was the best temperature, further supporting our data and hypothesis. The control variables we choose, a pH of 8 and 0.3 molars of sodium chloride salinity, resulted in the highest possible enzyme activity for our experiment. Group F from Section 11 tested enzyme activity of pH 5 through 11 and were able to determine that a pH of 8 was most optimal and proved that their values were all significantly different. Groups D and C of Section 11, tested salinity and found that 0.15 molar salinity had the highest enzyme activity, but their p-values were not able to show a significant difference. So the optimum salinity could not be determined in this case. Some sources of variability in our own experiment could be having multiple people pipette instead of just one, centrifuging some samples longer than others, and not putting the samples on the heat directly after the addition of trypsin. Some improvements we did make to our experiment between our first and second trial were: we changed our ranges of temperatures from 15 degrees Celsius to 50 degrees Celsius to 37 degrees Celsius to 80 degrees Celsius, giving us a wider range of temperatures. We also added controls for 37, 50, and 80 degrees Celsius, and instead of just running one sample in each condition, we ran 3. We had replicates this time. In future tests, some potential improvements could be focusing on a smaller range of temperatures closer to 50, running controls for every temperature tested rather than just the highest, middle, and lowest, and adding more samples at each temperature to further decrease variability. The ideal conditions to grow bacteria based on this experiment could be assumed to be temperatures between 45 degrees and 60 degrees Celsius, and a somewhat neutral pH, like the pH found in the human body. Our conclusion that 50 degrees Celsius, along with a pH of 8 being the optimal conditions for enzyme activity, are relevant in this assumption and therefore have a practical application. So this was our experiment done on trypsin. I have references listed down there. Thank you for listening.